Alright, hey AP Bio 2 kiddos, how you doing? It's Miss Lynham again, and this is going to be the first of several videos that I'm going to record over Unit 3, Cell Structure and Function. Remember, this is Chapters 4 and 5 in your Hillis book, so we're going to go ahead and get started and do a little short recording on the intro to cell structure and function, and then I'll have a couple other videos to follow this. Okay, so a little poem for you here. This is actually written by Mrs. Rice, so I have to give her credit for this, but it says, over 4 centuries ago, von Leeuwenhoek and Hooke stacked glass lenses together and took the first look at a world far too tiny for the naked eye to see and forever changed our understanding of biology. Isn't that so cute? I thought so too. Okay, so we're going to go ahead and start off with a little bit of history about microscopes. It's really hard to get you guys to understand about the microscope because you were born in this digital era, and you guys, from the time you guys were little, you guys had iPod touches and iPods and things like that, so it's hard for you to understand a world when we didn't even have a microscope. And I know in the classroom we have compound light microscopes that we use, and if you get a career or job in research, you also get to use electron microscopes. But now we have microscopes available, but if you look back to the time when Leeuwenhoek and Robert Hooke, and then there were other guys named Schleiden and Schwann and Virchow, they didn't have the advanced microscopy that we have available today. So the earliest microscope actually dates back to 1590, which was a very long time ago, but the earliest microscopes, the first ones, really just involved scientists stacking lenses on top of each other. And so the first guy that you see here, von Leeuwenhoek, okay, I'm going to write a little bit about him. Von Leeuwenhoek was the first scientist to use lenses and stack them on top of each other because it was several lenses, which kind of led to the compound microscope creation. He looked at pond water. Now, I know that you guys know and are very aware that we have things that are called protists that are found in pond water, but scientists didn't know that back then, in the early 1500s. So what von Leeuwenhoek did is he recorded in his lab journal and described what he saw as these little small organisms, and they were actually named animalcules. It sounds kind of funny, but they were really animalcules, which were kind of little small animals, little small organisms that he described, and of course we now know that these things were protists. Okay, after that, because once you have the discovery and scientists start recording their information in the journal, then other scientists want to come and repeat that experiment. So Robert Hooke didn't really repeat the pond water experiment, but he also took the first primitive microscope that was available because you have the invention of the first microscope after you have von Leeuwenhoek using lenses. So then the microscopes got a little bit better, and he looked at cork. Okay, now if you know a little bit about cork, it is actually bark from an oak tree. Okay, so bark from an oak tree. He looked at it. Here's what he saw, this picture up here, and he described what he saw as cells. He actually coined that name cell because he was a monk that lived in the monastery, and it was kind of like the nuns in the nunnery, or if you think of college students in a dorm room. Every room that is in that area is rectangular shaped and looks like the room next to it. So like that room looked like that room, which looked like that room and that room. And so in the monastery, they didn't call them rooms, they called them cells. And so he described what he saw as, hey, look at these little boxes in here. They look like the cell in which I live in, I reside in the monastery. And so he coined that name cell. It wasn't really anything scientific. It literally just meant like where he was living. Okay, so the two scientists that I just mentioned, von Leeuwenhoek and Robert Hooke, they contributed to the development of the cell theory, but there's some scientists that are not mentioned here as well that also led to the development of the cell theory. One of those, his name is Schleiden, S-C-H-L-E-I-D-E-N, and there's another one called Schwann. These two guys, Schleiden was a botanist and Schwann was a zoologist. And what they both did was started examining, like once Leeuwenhoek and Hooke opened the door to start talking about seeing things under these primitive microscopes, they decided to use the primitive microscopes as well. But Schleiden pretty much sampled like every plant he could find, whether it was, a small flower type plant or if it was a tree or mossy type stuff. He found every single plant he could find. He looked at all those plants under the microscope, and he kept seeing the same type of cells that Robert Hooke saw. And so Schwann decided, that the zoologist that he was, he started looking at different animal samples underneath the microscope. And so between Schleiden and Schwann's, their discovery, they literally said all plants and all animals are composed of cells. Leeuwenhoek said, hey, there's these little small animalcules that are in pond water. Robert Hooke sees these cells in cork. And so, of course, then we get to this guy named Virchow, Robert Virchow, that he looks under a microscope and he actually witnesses cell division. He saw a cell divide, and so the cell theory was created not just on these scientists that I have mentioned, but every scientist that led up to the discovery of the cell theory. It was their work that opened the door and let us create the cell theory, which says all living things are made of cells. So if it's alive, it's composed of cells. Okay, we already know that because that was one of the characteristics of life. But no matter whether it's a protist, a fungus, a bacteria, a plant, an animal, if it's alive, it's going to be made up of cells. Okay, the second part of the cell theory says the cell is the smallest working unit of life. Working unit of life. I like to say the cell is the smallest basic structure in all living things. Okay, it's the smallest that you can get that is alive, because I know you can go smaller than a cell and you have organelles that are inside of that, and then you have organelles that are made of molecules, etc. But the smallest working unit of life is the cell. That's the smallest you can get to have it be alive. And then the third statement is all cells come from pre-existing cells. Pre-existing cells. So that directly ties into Virchow's discovery when he saw cell division. That discovery from Virchow led to one of those statements in the cell theory, and we will talk a lot about that when we get to the mitosis, meiosis, and then even when we talk about binary fission, we will definitely talk a lot about the cells come from other cells. So the cell theory has those three statements: all living things are made of cells. The cell is the smallest basic unit of structure and function in all living things, or the smallest working unit of life, and all cells come from other cells. And once again, that was created by the work of all of these scientists. Okay? All right, the next thing is talking a little bit about microscopy, okay, or microscopes. I know that you are aware of at least the compound light microscope because we used that freshman year and then we're using it again this year. So the different types that you have are the compound light microscope, which you see here, and then electron microscopes, which we do not have at our school because they are very expensive. These electron microscopes were discovered in the 1950s, which was not that long ago, and that gave us a higher resolution power to be able to see things in a lot more detail. So if you look over here on the chart, this is a pretty good graphic where you can see the unaided eye will allow us to see, if you can see the measurements on the side, these are meters, and then you get to centimeters, millimeters, and then these are micrometers. So our human eye sees a little bit smaller than millimeters, so we are able to see frog eggs, of course bigger chicken eggs, human height, etc. That's with the unaided eye. So in this next spectrum, when you go from millimeters but then all the way down here to micrometers, you can see the modern light microscopes, compound light microscopes, and then some of them have super resolution, which goes down here to even be able to see like smallest bacteria. But then on this outside bar, these are electron microscopes, which obviously use a beam of electrons. That's what the word, that's what it means when they call it electron microscope. This will allow us to see all the way down to nanometers. You can see like little bitty small, like around this area, a little bit bigger than 1 nanometer. We can't get down to the size of seeing atoms, but we can see, let's say, the ribosomes, the different protein molecules, and certain lipids that make up parts of the cell. We can see that using these electron microscopes. So that's kind of how we discovered, you know, what things look like what that cell membrane looks like, is the ability using these electron microscopes. So over here on the side, if you look at compound light microscope, this is what we have in the classroom. There are a couple of areas, important parameters of those compound light microscopes. There's magnification, which is the ratio of objects image to size, to real size. The one in the classroom that we have, we can actually go up to 1,000 times the original size. This is an oil immersion lens. Okay, I'm just going to write it over here. It's an oil immersion. We will use it in our AP Bio classroom, but that literally, the 1,000 objective, 1,000 times, it's really a 100x objective, but the eyepiece, we can't forget the eyepiece, has a magnification in itself of 10 times. So if we use a 100 times objective plus the eyepiece is 10 times, that gives us the total magnification, which is 1,000 times the original size. That's pretty powerful. So that's one aspect of the compound light microscope. For us in the classroom, we're also going to use a 10x objective, which is our low power, and then we have a 40x, which is our high power. So I like to call the 40x is high, 10x is low, and then we have a super high, which is the 100 oil immersion lens. Okay, the second aspect that you can look at with a microscope is the resolution. It's basically the clarity of the image. It kind of is like basically the minimum distance between 2 points being separated and still being seen. That gives you clarity of that. On our microscopes, they go pretty much from 0.2 micrometers all the way up to or 200 nanometers is where we get on that. Okay, the advances in light microscopy, this is, like current advancement. They now look at things like the confocal ability. They look at the fluorescence of the microscope. They can do phase contrasting. They can do super resolution. So there's a lot of advances in this area with technology involved in that to try to get better and better with microscopes. So it's kind of cool when you start looking at that. The bottom section, the electron microscopes, the two that I want you to know are SEM and TEM, and just as the word implies, it shines a beam of electrons through the object, through the specimen. It can achieve a resolution of 0.002 nanometers, which is pretty powerful. It's basically it's a 100-fold improvement over the compound light microscopes. So it's pretty amazing that it can do that. The only drawback actually to an electron microscope is the specimen must be kept in a vacuum. Okay, so the specimen is put in a vacuum tube, and what that does is kills the specimen, so you're not going to be able to have like a live specimen, whereas with a compound light microscope, you could take a sample from pond water and watch living things move underneath the microscope and then release them back to the pond. So this electron microscopy does have a disadvantage because it kills the specimen. So that's one thing I wanted to mention. But the difference between the two, if I could just erase this and then put it back up here, the difference between the two electron microscopes, if you look at the SEM, SEM means scanning, T means transmission, the EM is electron microscope. So the scanning will allow you to study the topography of a specimen. It scans the surface, okay, scans the surface of the sample. Usually what you would see is you could see like the outer surface of the cell membrane because those beams of electrons will go across the surface. You can see a lot of detail, but it would allow you to see surface proteins or see across the phospholipid bilayer, whereas transmission electron microscope would give you three-dimensional appearance of the internal organelles. So if you wanted to see, let's say, the cristae of the mitochondria, you would use this TEM because you would be able to see through the mitochondria to visualize those internal organelles. So that would give you a little bit of a better advantage on that. Okay, next thing we're going to talk about is the cell size. So we're going to take a little break for a minute. Okay, now we're going to talk about cell size. Did you ever wonder about the size of cells? Like, do elephants have ginormous cells compared to humans? Hopefully you realize that cells must be small. Doesn't matter whether it's an elephant, a giraffe, a human, a bacteria. The size of a cell must remain small because there are some metabolic needs that impose both upper and lower limits on the size of a cell. So the first thing I want to do is just tell you that they do range. There are some ranges in size. A typical bacteria, okay, a typical bacteria which might be E. coli or Salmonella, etc., is between 1 and 5 micrometers. That's the typical bacterial size. Okay, now there are some bacteria that are called mycoplasma. You can see them down here. They are less than 1 micrometer. In fact, they range between 0.1 and 1, depending on the specific example. So that's pretty darn small. A typical eukaryotic cell, which is our cells, are between 10 and 100 micrometers. So there is a varying range. Bacterial cells are definitely smaller than eukaryotic cells. So they're going to have an advantage as far as getting things in and out of the cell at a faster rate. The smaller the size, the faster the rate is going to be able to get things in and out. But there is a range, but all cells are pretty much on the small side. Okay, now first thing we want to look at is this surface area to volume ratio. We can actually use a model in the classroom, which we're going to do this week, to study the surface area versus the volume ratio in a cell. So if I draw a typical cell, here you go. You guys know that the outside is called the what? Okay, hopefully you know cell membrane. Okay, so the cell membrane is the structure that regulates homeostasis by regulating what's going to go in and what's going to go out of the cell. That's the homeostatic function of that. So the surface area is representing, or the cell membrane represents the surface area of the cell. Okay, the volume is everything that's on the inside. So that's the cytoplasm. That would include the cell gel, plus all the organelles and everything on the inside of this. So if you were to imagine this cell would grow in size, like if you doubled the size of this cell, if you mathematically put some numbers to this, you would actually increase the volume at a faster rate. Okay, so the stuff on the inside would increase at a faster rate than what that surface area would be able to support. So therefore, that cell, it's physically not possible for it to keep growing, growing, growing and have unlimited growth. Instead, what organisms do, like in the case of us, is our cell might increase a little bit in size, and then it's going to send a signal for it to divide. It only grows slightly in size, and then the cell will divide into 2 cells so that our cells stay small to be able to get materials in and out. Okay, so I wanted to mention also the volume, if my pen will work, the volume of a cell, okay, right over here, determines the amount of metabolic activity that it carries out per unit of time. So for instance, inside the cytoplasm, you've got the mitochondria that's going through cell respiration. You've got, the ribosomes that are producing proteins. So the volume determines the amount of metabolic activity that's going on per unit of time, whereas the surface area determines the amount of substances that can enter or leave the cell from the outside environment, and like how many waste products would leave from the inside to go to the outside. So those are metabolic needs that a cell must have. Now as far as like how small can it be, there has to be enough space in that cell for DNA to fit, enzymes, and even if it's a small microplasmic bacteria, they do still have enzymes and they have their DNA in a nucleoid region, so it's got to be big enough to support that. Okay, so the cell has to have enough surface area, that cell membrane, to support the cell's metabolic needs. But if you were, like I said, if you look at this chart, if you increase the cell in size, and they used a mathematical cube to represent this, we know our cells are not cuboidal shaped, but if you use the surface area to volume rule and you try to figure out what would happen if you went from a cube, a 1 by 1 by 1, to a cube that was 5 by 5 by 5, then you would see that the surface area would increase, or excuse me, the volume would increase at a faster rate than what the surface area would be able to support. Now we're going to do a lab in this, in the class this week that's going to illustrate this with some gel cubes, so you're going to be able to actually witness this. But the one thing I want you to write down right here, put a little star, is that as the cell increases in size, okay, go ahead and write this down, as the cell increases in size, oh my pen, okay, as cell increases in size, comma, the volume grows proportionately more than the surface area. Okay, so as the cell increases in size, the volume, which is everything on the inside, the cytoplasm, grows proportionately more than the surface area. Okay, so you're going to be able to see this. Now, what is good, what is beneficial, is organisms want a high surface area to volume ratio. One that is high, it's a high surface area to volume ratio, facilitates the exchange of materials between the cell and the environment. So if you look at this diagram, this little bitty teeny tiny cube, if you look at the surface area to volume ratio, the surface area is 6, the volume is 1, and all we did was use our formula, height times width times length, the number of boxes, to find the volume, and then surface area, we took surface area to figure that. So this is a 6-to-1 surface area to volume ratio. That is a large surface area to volume ratio. That's good, because that means that, like I said, the smaller the size, the more easily substances can move from one location to another, like within the cell and then also to move out of the cell. And I always like to think, if you had this humongous cell that was really, really big, and you had a mitochondria right in here, and you needed to get glucose into the mitochondria, if that cell was huge, think about the amount of time that it would take for that glucose to get into that to reach the mitochondria, whereas if you had a tiny cell, there's the mitochondria, glucose would be able to get in faster. So the smaller the cell, the more beneficial, because it has a higher surface area to volume ratio, which allows substances to easily move from one location to another within that cell. Okay? Does that make sense? Sweet.